Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were discussing cancer biology, in which we learned about different hallmarks of cancer. So, in last class, we learned about angiogenesis, which is quite important for ensuring the constant supply of oxygen and nutrients to the uncontrollably growing tumor. Okay, so today we will be learning about regulation of an important factor, the regulatory pathway for that factor, which helps in promoting angiogenesis. So I have already mentioned this factor in last class. So today we will be learning about regulation of HIF-1 alpha, the hypoxia inducible factor 1 alpha. So the regulation of this factor under the normal condition means when the oxygen levels in the tissue is normal, so that we refer as normoxia, and when the oxygen levels in tissue is low means hypoxia. So we are going to see when in which case it gets degraded and what kind of pathway is followed so that the HIF-1 alpha can do its downstream signaling. So under normal conditions, this HIF-1 alpha, this is hypoxia inducible factor. So it is going to activate the genes which are important for a tissue to for the tissue survival under the hypoxia conditions, and that is why the name is hypoxia inducible factor. So this is induced whenever there is hypoxic conditions, means the cells are they are thriving under the low oxygen level. So whenever there is low oxygen, you need to do certain changes in the tissue so that the tissue survives. For example, you have to promote angiogenesis, which will ensure that the tissue gets enough oxygen to survive. So though those genes, the genes which are important for a tissue to survive under the hypoxic condition, that will be activated by HIF-1 alpha. So whenever there is normal level of oxygen, you need to deactivate or inhibit whatever genes they will be controlled by the HIF-1 alpha. So for that matter, you need to degrade the HIF-1 alpha whenever there is a normal condition. So that is why in normoxia conditions, the HIF-1 alpha, it undergoes degradation. Okay, and for undergoing degradation, first it undergoes hydroxylation. Okay, the hydroxylation of HIF-1 alpha is done by the enzymes known as prolyl hydroxylase domain containing enzymes, PHDs. Okay, these are prolyl hydroxylase and domain containing enzymes. So these enzymes hydroxylates using oxygen, they hydroxylates the proline residues that are present on the HIF-1 alpha. So this hydroxylation, this marks for this marks the HIF-1 alpha for the recognition by a protein known as von Hippel-Lindau protein. Okay, so due to the hydroxylation, the HIF-1 alpha can be recognized by a protein known as von Hippel-Lindau protein. Okay, so this protein is a component of E3 ubiquitin ligase complex. Okay, so once the PVHL, this protein, this recognizes the HIF-1 alpha, it is as it is a component of E3 ubiquitin ligase. So it is component of this complex, and so this is going to promote ubiquitination of the HIF-1 alpha, and we already know that ubiquitination is a mark for the protein degradation. It suggests that this protein is meant to be degraded. So once the HIF-1 alpha is ubiquitinated, it undergoes proteasomal degradation. After ubiquitination, it undergoes proteasomal degradation. And as the HIF-1 alpha is degraded, this prevents the accumulation of this factor. And as the HIF-1 alpha is not there, so no hypoxia related genes will be activated. So this is what happens under normal conditions. Under the hypoxia conditions, instead of degradation, HIF-1 alpha is stabilized. Okay, so stabilization of HIF-1 alpha is done. So how is it done? So during hypoxic condition, the activity of the prolyl, sorry, not the activity of prolyl hydroxylase enzymes, the PHD enzymes, this is this is inhibited because in the low oxygen level, during the low oxygen they cannot work. So once as the PHD enzymes, they cannot hydroxylate HIF-1 alpha, then the HIF-1 alpha is stabilized, and due to the stabilization of HIF-1 alpha, it gets accumulated in the cytosol. Once it gets accumulated in a certain amount, it gets translocated to nucleus. So then it gets translocated to the nucleus. Okay, so when it comes in the nucleus, at this point, the HIF-1 alpha, it forms a dimeric complex with HIF-1 beta. Okay, so this dimeric complex, this binds to the hypoxia responsive elements. This is the DNA domain. Okay, the part of the DNA which is found in the promoter region of the target genes, the genes which are meant to be get activated under the hypoxic conditions. So this activates the transcription, and this helps the interaction with certain cofactors which are going to enhance the transcriptional activity, the co-activators like P300. Okay, so now the transcription will be seen, and the downstream genes which are important for which are important under hypoxic conditions will be the growth factors like VEGF, the vascular endothelial growth factor, which is going to promote angiogenesis. Then the other growth factor like platelet-derived growth factor, the matrix metalloproteinases, as well as angiopoietin. Okay, so let's summarize whole scenario. What happens under normoxia and hypoxia? Okay, so in both of the factors, the HIF-1 alpha is present. In both of the cases, HIF-1 alpha is present. Okay, so I'm going to show HIF-1 alpha with this blue color. Okay, so what happens in normoxia, this HIF-1 alpha, on this HIF-1 alpha works the PHD enzymes, the hydroxylase enzymes, okay, and they do the hydroxylation of the okay. So they have done the hydroxylation of HIF-1 alpha, and due to the hydroxylation of HIF-1 alpha, now this gets recognized by this hydroxylated HIF-1 alpha. This gets recognized by the protein known as von Hippel-Lindau. Okay, so this protein recognizes this HIF factor Okay, and once due to the recognition, this is the this is part of the E3 ubiquitin ligase. So now this E3 ubiquitin ligase will do the ubiquitination, and due to ubiquitination, you will see the proteasomal degradation. So the HIF-1α, it is degraded into the proteasome. Okay, so, but there are certain other factors, some other factors which are going to, instead of doing the hydroxylation of proline residue, so PHD enzymes, they do the hydroxylation of proline residue, but instead of doing the hydroxylation of those residue, the hydroxylation of asparagine residue is also done by some other factors, by some other enzymes. And due to the so due to the this hydroxylation, this doesn't undergo the proteasomal degradation, but hydroxylation of asparagine residue, this prevents the interaction with certain co-activators that I mentioned previously, the co-activators like P300. So now P300, they cannot interact with HIF-1 alpha, so there will be no downstream signaling. So the HIF-1 alpha cannot bind to the, it cannot dimerize, and it cannot bind to the hypoxia responsive elements. So there will be no transcription of the growth factors. Okay. But under the hypoxia condition, this this HIF-1 alpha gets stabilized. So that whenever there will be low oxygen, HIF-1 alpha will be stabilized. So the PHD, they cannot work, and due to this PHD cannot work, this HIF-1 alpha comes in the nucleus. So this is the nucleus, and in the nucleus, this HIF-1 alpha gets dimerized with HIF-1 beta. So this is the HIF-1 alpha HIF-1 beta complex. Okay, so this complex can now go and bind to the hypoxia responsive element. So this is the DNA. Okay, and here is the hypoxia responsive element. So this part of the DNA is the hypoxia responsive element, which is present in the promoter region of the growth factors. So now this, the once this binds to the hypoxia responsive elements, this complex binds to the hypoxia responsive element, it it interacts with the certain factors like P300, which are the co-activators of this pathway. So now this is going to enhance the transcription of the growth factors like vascular endothelial growth factor, which is going to promote angiogenesis, or the platelet derived growth factor, which is going to support the cell survival and proliferation. So now the cells can survive under low oxygen conditions as well. Then it also activate the transcribes the matrix metalloproteinases, which are going to facilitate the tissue remodeling, which is quite important under the hypoxia conditions. Then it is also going to upregulate angiopoietin. Okay, this is critical for blood vessel maturation and stability. So this is going to provide the stability to blood vessels. Okay, so all of those expression of all of those factors, it is quite important for the survival of the cells under the hypoxic conditions. Okay, so this is how the HIF-1 factor is regulated under the normal and hypoxic conditions. There are certain important why do we upregulate the HIF-1 alpha? Because it is going to increase the angiogenesis, ensuring the oxygen supply to hypoxic tissue. But beside this, it has other roles as well, which I did not discuss in the previous lecture. For example, it enhances glycolysis. And why is it important? Because enhancing glycolytic pathway, it is going to allow the ATP production. So the ATP is quite important under the low oxygen conditions, so that the metabolic activity it ensures that the whatever metabolism should is important for cell survival, it will go on, it will continue to happen. And then it also promotes erythropoiesis. Okay. HIF-1 alpha, when it gets activated, it enhances angiogenesis, enhances glycolysis, and it promotes erythropoiesis. This increases the oxygen carrying capacity of blood. So now even if the oxygen concentration is low in the surrounding tissue, but due to the increase in oxygen carrying capacity of the blood, now the hypoxic condition can be normalized. Okay, so this is what happens in the hypoxic condition, and that is why HIF-1 is upregulated. But in cancer condition, tumor cell exploit this pathway to survive in hypoxic environments, because now in the normal tissue, the certain cells are going uncontrollably, so they are depriving other cells of the nutrients. So tumor cells, when they uncontrollably grow in certain environment, the the blood which was providing normal oxygen levels, now it is not able to provide whatever oxygen it was providing to the tissue in the normal conditions, because the number of cells is increasing, and that is creating hypoxic microenvironment. And in this hypoxic microenvironment, tumor cell exploit this pathway. They promote, they upregulate the HIF-1 alpha, so they accumulate HIF-1 alpha in the cell, and they promote angiogenesis. So due to the angiogenesis, now the constant blood supply will be maintained, which will ensure the constant supply of oxygen as well as other nutrients which are helpful for the uncontrollable growth. And this will now increase the other, this will increase the chances of the tissue being the metastatic, having the metastasizing capabilities. So this will, promoting angiogenesis in the tumor cases, this will lead the harmless tumors. This will lead the progression of harmless tumors to harmful tumors, because due to the angiogenesis, now the tumor cells, they are going to acquire the capability of metastasis. Okay, then this happens in cancer, but in other diseases like the pulmonary disorders, where the chronic hypoxia induced HIF-1 activation, it is going to contribute to the pulmonary hypertension. Then in other diseases like ischemic diseases, HIF-1 activation is quite important, because it can aid to recovery. It can aid to recovery in the stroke or myocardial infarction by stimulating blood vessel growth. So now as the angiogenesis is promoted in ischemic diseases, it can recover the tissue uh, by providing the normal supply of oxygen. So hypoxia, HIF-1 alpha, it is not upregulated in the hypoxic condition only, but it is upregulated in the cancerous condition, when the tumor cells start proliferating in the ischemic diseases, where it is going to aid to recovery. This is also, upregulated in the pulmonary disorders, because in the pulmonary disorders, there will be chronic hypoxia. Okay, and that is going to activate the HIF-1 alpha, and this is going to contribute to the pulmonary hypertension. So with this, we are done with the regulatory pathways of HIF-1 alpha. We have seen how the dysregulation of this system plays a pivotal role in the cancer progression, and how the HIF-1 alpha is quite important in recovery in certain diseases. So with this, I end the lecture. In next lecture, we will learn about other hallmarks of cancer. So till then, see you. Bye-bye.